So we'll continue the subject of CMT, the CMT section, and our next speaker for today is Dr. Trifa, who will talk about inheritance and genetic aspects of CMT.
So, hello, thank you for the kind invitation, and I am very honored to be with you today and to talk about some genetic aspects regarding this very interesting disease, which is Charcot-Marie-Tooth. So I am associate professor of genetics at the University of Medicine and Pharmacy from Cluj-Napoca, Romania, and I also work in a cancer institute. Actually, I worked a lot on cancer, on genetic aspects regarding human cancers, and then I started to have a special focus on neurological genetic diseases, included CMT. And I also am the president of a humanitarian association, which is called Noi pentru ei, NEI, and we help patients with genetic diseases by this non-governmental organization. So I would like to point out some basic concepts that we use in genetics. Actually, these concepts are not specific to Charcot-Marie-Tooth, but they can be applied to all of the genetic diseases, all of the human genetic diseases, which are in a large number. We currently know more than 8,000 different genetic diseases in humans, and that's really impressive. A lot of them affect the nervous system. So some basic terms that we hear a lot of times. The chromosomes. The chromosomes represent the support of the genetic information, and all of the genes, and in general our genetic information can be found on these chromosomes. And we have 46 chromosomes, and they are grouped by pairs. One comes from the mother and the other comes from the father. And of course we have not only the autosomes, but we have the sex chromosomes, the X and Y, which are specific and determine our genetic sex and our external aspect. So the chromosomes are made of DNA and some specific proteins. Most of the human DNA can be found in the nucleus of the cells. We are built. We have trillions of cells in our body, and most of these cells have a part of these cells have the ability of renewal, and this process of renewal supposes also the multiplication of the DNA. So DNA is a long molecule. It has almost 2 meters, and we can imagine that these 2 meters have to fit the limited space of the cell. So the DNA becomes condensed under the form of chromosomes in order to fit the limited space of the nucleus. We have DNA not only in but other parts of the cells that are called mitochondria, and these mitochondria are very important for the production of energy, of ATP. All that we do, all actions we perform in our daily life suppose the formation of this energy, which is produced by these components of the cells. So the genes, what are the genes? The genes are stretches of parts of DNA, and we have about 20,000 genes, and each gene codes for a specific protein, but not only proteins, but we have different other types of molecules that are coded by these genes. So we can say basically that a gene codes for a specific biological function, or controls a specific biological functions that takes place in our body. So actually, what is the most interesting, the genes represent only a small part of the genome. So the genome is all of the information, the genetic information we have in our cell. That's the genome. Now, if we talk about genetic diseases, then we'll be interested mostly in these genes. In general, if we have a genetic disease, including Charcot-Marie-Tooth, that means we have a corresponding or more corresponding genes that suffered some modifications, because the genes are formed from some bricks that we call nucleotides, and our genetic code contains 4 types of these letters, which we called C, that means cytosine, A, adenine, G, guanine, and T, thymine. These are the 4 letters that define our genome. So we have 4 letters, and in each gene contains a group of these 4 types of letters in a specific order, and that defines the function of the specific function of that gene. So for each gene we have 2 alleles. So the alleles represent the variants of the same gene. So in a given population, we may have a lot of alleles for the same gene, because that's the evolution that produced these multiple alleles for a specific gene. But for each specific gene, we have 2 alleles, and we inherit one of them from our mother and the other from our father. So each one of our parents contributes with a copy for each gene, except for the mitochondria, which is inherited from the mother. So the genotype defines the combination of the 2 specific alleles that we have for each gene from our genome. The phenotype is what we can observe at the exterior. So the phenotype is the result of the interaction between the genotype, so the genetic information, and the environment. So each one has a different phenotype because there are small differences in our genomes. Each genetic disease has a specific phenotype due to specific modifications in specific genes, and that is the case of Charcot-Marie-Tooth. And as Professor Adrian talked about, Charcot-Marie-Tooth is a very heterogeneous disease from the genetic point of view. There are a lot of genes and involved in the apparition of Charcot-Marie-Tooth disease. Now, a really important are these three terms: homozygous genotype, heterozygous genotype, and hemizygous genotype. Homozygous genotype means that we have the same alleles that are identical. So homozygous means that we inherited two identical alleles from our parents. If these two alleles contains one mutation, each one of them, that means that in the end we will have two copies from the mutation. Heterozygous genotypes means that we have two different alleles. So we inherited one allele from the mother, let's say, and one different allele from the father. Hemizygous genotypes applies only for the genes from the X chromosome, because in men there is one X and one Y. There is no correspondence. In general, there is no corresponding genes on the Y chromosomes in men. So men who inherit mutations on the X chromosomes, they will be hemizygous for those mutations. So how genetic diseases are inherited in general, and Charcot-Marie-Tooth is no exception. So we have autosomal inheritance. That means that the mutations are found on the autosomes. That means on chromosomes different from X and Y. And in this case, we can have dominant or recessive inheritance. And dominant means that one copy of the mutations is enough for having the disease. So if we inherit of the mutation is enough in order to have the disease, and we will say this is a autosomal dominant transmission. Autosomal recessive means that we need two copies from a mutation in order to have the disease. So both parents should give us one mutation, each one of us. So they are carriers in this case. So we need two copies of the mutation in order to have the disease. There is also X-linked inheritance, and in this case, the mutation is found in a gene from the X chromosome. And in this case, if the mother is carrier, for instance, if the mother is carrier of the mutation, she has two X chromosomes, so she will not have the disease in general, but she can transmit it to the to the children, for instance, and mostly to the boys. So if a boy inherits a mutation from the X chromosome from his mother, that means he will have the disease because he has no X chromosome to compensate for the mutation. This X-linked inheritance two nuances. That means we have a dominant X-linked inheritance and recessive X-linked inheritance. And if a disease is X-linked dominant, usually it affects not only boys, but also girls. In the same time, X-linked recessive disease affect most of the cases only the boys. Because, like I said, in this case, if a girl inherits one mutation from the X chromosome, she will have the other X chromosome that will compensate for the mutation. And we have specific forms of Charcot-Marie-Tooth disease that are transmitted from the X chromosome. They have an X-linked inheritance. This is the case of JGB1 gene, for instance, that produces Charcot-Marie-Tooth 1X subtype of disease. So, like Professor Adrian told us a few minutes ago, it is possible to have sporadic or de novo mutations, and in this case, our parents are neither carriers nor ill individuals. Or in this case, the mutations appear either in spermatozoa or in ovum of our parents, or after the fecundation has taken place. So in this case, we will say that we have a de novo or sporadic mutation. This for some of us, for some of the of the people that have the disease, this may seem paradoxical. No one in the family has had the disease, and why me? Why do I have the disease? Because nobody before me in the family has had it. And it's sometimes I've noticed frustrating for them not to have individuals that have the disease in the family. So, Charcot-Marie-Tooth, one of the most frequent genetic diseases, one of the most, if not the most frequent neurological diseases. For sure, it is the most frequent hereditary neuropathy. And I will not insist on the clinical aspects of the disease because Professor Adrian presented us very nice all this data. I want to talk about the genetic heterogeneity that we have in this disease, and which led to the proposal of a new classification of the disease. So we know the traditional classification in demyelinating or CMT type 1, and axonal CMT type 2. And most of these forms are being transmitted in a dominant fashion. So this classification appeared in a moment when we had no information, we had no clear information regarding the genes, regarding the sequence of the genes, the sequence of the genome. So we had only the electroneurophysiological informations and the clinical picture of the patient and the inheritance mode in the family. So other terms were coined out at that time, and are being part of the traditional classification, which is dominant intermediate, who is characterized by intermediate velocities for the nervous impulse, and Dejerin-Sotas syndrome, which was called CMT type 3, but this is a rather ambiguous term because all of the severe forms of CMT could be included in this particular type. And CMT type 4, which refers to recessive forms. But after that, the genes responsible for Charcot-Marie-Tooth have been discovered one after the other, and this complicated a little bit the picture. And after that, the various types from the traditional classification started to earn letters. So 1A, 1B, 1C, 2A, 2B, and etc. So for type 1, for instance, other subtypes appeared. 1A, which is the most frequent, and this is a duplication of the gene PMP22, and this particular type represents about 30-60% of all CMT patients, depending of the ethnic origin of the patients. GGB1, which produces Charcot-Marie-Tooth, X-linked Charcot-Marie-Tooth or 1X, CMT1B due to mutations in the MPZ gene, and a lot of other subtypes that have been called after the discovery of their specific genes. And for type 2 CMT, it's the same case. We have the most frequent gene, which is the mitofusin 2, which produces CMT2A subtype, but a lot of other genes have been discovered that produce this particular type. So most of them are being transmitted dominant. I said that Dejerin-Sotas syndrome, or CMT type 3, is a rather ambiguous term because several different genes can produce this particular type of CMT. And in my opinion, this term should be avoided due to these possible confusions. And a lot of genes involved in the CMT type 4, which is recessive. Some of them are demyelinating, and some of them are axonal. So a new proposal, a new classification have been proposed due to this important number of genes that emerged in the last year, and a lot of letters, a lot of genes. So there is this simplified proposal, this new classification, which contains several informations, such as the mode of inheritance, autosomal dominant, autosomal recessive, X-linked, sporadic, and etc. Then CMT, Charcot-Marie-Tooth. Then the major type, which is axonal, demyelinating, or intermediate, and the gene discovered after we performed the genetic test. So one example, this type, which is the most common, CMT1A, due to mutation, due to the duplication of the PMP22 gene, called autosomal dominant, CMT DA, so demyelinating, and PMP22 in the new classification, the new code of this subtype. Even more complicated, this gene GDAP1, which produces several different subtypes in the old classification, and this could be translated in the right under the new classification. For instance, autosomal recessive axonal GDAP1 related subtype corresponds to CMT2H in the traditional classification, and so on. We know the phenotype of the patient. We know the electrophysiological aspect of the transmission of the nervous influx. We know the mode of inheritance in the family, and we are able to perform nowadays all the genetic tests, not all, but a great deal of genetic tests, and we have the gene, and we can classify more in a more easier way, in my opinion, each particular patient. We can assign him to a specific subtype of the disease. So very important nowadays. We had no information. We had no I don't know, 40, 50 years ago. So we were not able to analyze the genes involved in CMT, but nowadays we can do that. So the first tier test that we perform, especially if it's about a demyelinating patient, is a PMP22 duplication, which is responsible for the greatest deal of patients of CMT. If this is negative, or we can go forward to a multi-panel gene testing by next generation sequencing. This is a technique that evolved a lot in the last 20 years, and this allows us to analyze simultaneously most of the genes currently known to be involved in Charcot-Marie-Tooth. We can do this directly if we have an axonal or an intermediate phenotype. If we find a mutation, this way we can assign the patient to a specific CMT subtype. If not, we can perform whole exome sequencing, that is sequencing of our genes, or whole genome sequencing. Apparently, there is no advantage of one over the other. If we find a mutation, okay, we can assign the patient to a specific CMT subtype. If not, and this is the case for 10 to 30% of the patients, well, we performed, after performing all the genetic tests available these days, this is still the case for a part of the patients in which we still can't find any mutation. A problem that we encounter currently in genetic testing is VUS. This VUS is a variant of undetermined significance, and this is one of the major drawbacks of the the genetic testing in Charcot-Marie-Tooth, because these are variants of uncertain significance. These are mutations that perhaps have not been described before, and we don't know if they are pathological or not. Maybe their status will change in the future years, so we should take into account this possibility. Another problem regarding the genetic tests, the genetic testing in Charcot-Marie-Tooth, is that a lot of patients will need complex genetic tests, such as next generation sequencing panels or whole exome sequencing, and etc., which in these days are becoming more and more widely available and more affordable. And very important, a part of the patients remain, after performing all of these tests, remain without identifiable mutation. And the lack of specific treatment represents also a problem, but hopefully in the next years we will have some options from this point of view. And now I would like to point out something that we see in Romania, because I practice in Romania. In our country, although we expect to have several thousand of patients, a small number of them have the genetic test, and this is very frustrating for me as a geneticist, because sometimes neurologists they don't communicate with us. They don't refer the patient to us, to the geneticist, and the patient remain without genetic tests, and that's really annoying for me. And we still don't have in Romania a national registry for Charcot-Marie-Tooth disease. So hopefully this problem will be solved in the next year. And in the end, I would like to thank you for the opportunity you gave me to talk at this wonderful conference. And this is footage from my home city, which is Cluj-Napoca, very beautiful city, the capital of Transylvania. And hopefully we'll see each other on site after in better times.
Thank you so much. I have to tell you that I've had the pleasure of visiting Cluj-Napoca for a rare disease conference some 8 years ago. So it's a beautiful city, and I think the university there is really at the avant-garde of so much research on rare diseases. So
Yes.
I'll envy you, I guess. There are some questions that concern the mutations. So questions from the audience are how is it possible that you have a single gene and in the phenotype you see so many different types of CMT?
Yes, that's intriguing for the geneticist as well. The genes code for specific proteins, and each of these specific protein has in multiple parts, and each part of this protein has sometimes different roles. So if we have a mutation in this part of the gene, we'll have a phenotype. If we have another mutation in a different part of the gene, we may have a different phenotype. So mutations in the same gene that produce different diseases, different phenotypes.
Right, so we are having short, genetic classes here, it seems, because the next question also concerns the understanding of genetics. So the question is, could you explain how about the variants of the underdetermined significance that can sometimes be found in patients with CMT?
Well, that's quite a big problem that we have sometimes this variant of undetermined significance that sometimes this is very tricky. So this type of modifications, this variant of undetermined significance, maybe in a few years they will become true mutations, or they will remain variants of undetermined significance. In general, if we identify such a modification, we should perform more tests in order to see the effects of that particular variant. That could enable us to reassign its effect.
All right. So thank you so much.